The presentation is going to be more like myself thoughts really on the issue of the use of CT data to measure deformation, which is my main interest, and also to go from deformation to material identification. So I'm a professor in engineering in Southampton. I've been in Southampton for the last 8 years now. And my main interest really is on the design of novel test methods for materials based on full field deformation measurement. Initially, obviously, with standard cameras, CCD, CMOS, but also volume measurement is of particular interest. And this is what is on for today's talk. So a brief outline of my talk. I will start with a brief introduction about motivation, and I will say a few words about digital volume correlation, which I'm sure a lot of you around this virtual table know about. I will then show a few examples based on my own research. That was simply because it was easier to get images, but there are a lot of papers out there. And what I think will be the most interesting part of the talk really is the reflections about what I call the DVC curse. I'll explain about that a bit later, and how we might be able to exercise that curse. And this is a bit more speculative and looking forward and really to fuel the discussions. So let's get started with the introduction and DVC. So, I mean, we all know that full field deformation measurements have made a lot of progress within, I would say, the last 20 years. First, because of the progress in imaging technology, mostly initially CCD and CMOS cameras, but also the progress in computing power and the fact that we're able now to do very advanced data processing of images we might collect. And so we can measure deformation at the surface. This is a video from my research group showing a strain wave in a flat specimen impacted, and you see the time incrementing in microseconds. So some of these cameras are very fast. Oops, sorry. But with other imaging protocols, we can actually go to volume measurements. And I'll be obviously talking about that a little bit more because that's the focus of the presentation today. So there are different ways of measuring, or rather making images in the volume. I will just cite a few standard or maybe less standard, some of them protocols. Confocal microscopy you all know about. That's really for very small scales, mostly in the biomedical field. As is optical coherence tomography, which is another thing you might have heard of, which is very interesting as well for biological tissues. Magnetic resonance imaging, and also a derivative of MRI called magnetic resonance elastography, which provides directly 3D maps of displacements with very nice resolution. And this is an area that is less well known than MRI. Ultrasound imaging and ultrasound elastography meaning the capacity to image the elasticity of materials, meaning stiffnesses, and generally what people there are interested in is contrast of stiffnesses. Just for the record, colleagues in Loughborough a few years back developed a very nice method called tilt scanning interferometry, and there are different variations around that theme, which could provide 3D displacement maps inside semi-transparent solids. Very nice work. But what we are concerned with today is X-ray computed tomography, which I think is certainly in engineering the most popular way of generating volume images. So I will not introduce you to X-ray tomography. That would be a waste of time. I think everyone around the table is very familiar with all this. So basically what you get is a volume image. I will call this an image, but it's a volume image. And it's an image that contains contrast based on generally absorption. So differential absorptions between different constituents in the medium that's investigated. So that's a typical image that you could get. You could see sort of black and white contrast, different gray levels that could come from the microstructure of what is imaged. And in the late 90s, researchers came up with an extension of a technique called digital image correlation that tracks movements at the surface of solids, extension to volumes, and that was christened digital volume correlation. Now, if you can imagine, this is the volume before load is applied, then you can apply mechanical loading in situ. There are a number now of in situ loading rigs, and you will get another volume after deformation. Now, what you would like to do ideally is to consider a voxel in the initial volume and see where it's moved in the deformed one. Unfortunately, because of digitization, a voxel with a particular Bray level will have a lot of counterparts in the deformed volume because of the limited number of Gray levels. So a voxel's Gray level is not enough of a unique signature to be able to do this tracking. So the standard way around this is, instead of considering one voxel, we consider a subset of voxels that I represent here. And if this subset is large enough, then it presents a unique signature that you can track now between the undeformed and the deformed configuration. So once you've done that, then you can build up a displacement vector between these two volumes, and you can then scan, raster this little subset all over your volume and get a field of deformation, a field of displacement in the bulk and in 3D. So how does it work in practice? Well, in practice, what we write is the optical flow conservation. And so if you consider that F is your initial undeformed image and G is your deformed image after loading, then the matching consists in identifying a displacement field, U, of in each position. X is space variables, so that the deformed image of X plus U matches the initial image F. And so you build up a least square problem with that, and the solution is the minimum of that function here. Now, to do that, you need to parameterize the displacement. And this is where a lot of the problems of DVC can come from. I will illustrate in a few minutes. If you consider just 2D DIC, surface DIC, but it's exactly the same in 3D, it's just easier to write on the board. You can first write the location, the coordinates of the points after deformation as the initial coordinate xy plus the displacement of that point, u and v. If you are tracking u and v, it means you are considering that your subset is not deforming through the transformation. So over that little scale, you assume that it's displacing as a rigid body. And so you can identify these two values, u and v. That's the lowest order parameterization you can potentially think of. You can enrich that base. If you have slightly larger deformation, you might say that actually this little subset will also deform. And if you want to track the displacements better, you will need to add a higher order part to this shape function. So you could go for affine. And in that case, you have now also the derivatives here to identify. So you now have to identify more parameters in this matching, but this will give you a better estimate of the displacement of the center of the subset. And you can go for higher shape functions, particularly quadratic, and it's shown in the literature for DIC that quadratic is actually the optimal parameterization for the displacement in this matching problem. And you have more information to identify. So in the slightly larger subset, but this is generally how it's done now in commercial packages. The alternative parameterization, you could use a mesh, a bit like an FE mesh, so have a piecewise parameterization, or you can have closed form parameterization, for instance, in fracture mechanics. If you know the K-dominated solution of the displacement ahead of a crack tip, you might want to project your DVC data onto that kind of basis. And there are many others. It's a very large playing field. And in the later part of my talk, I will present an application where beam kinematics are used for this parameterization. So that's just a brief introduction about digital volume correlation. I'm just going to now give brief examples of application, just as illustrations, really, before starting the more interesting part of the talk, I think. So this is the first time I manipulated X-ray CT data. It was a collaboration with the University of Manchester at the time with Phil Withers, and Andrew Alderson was working on Auxetic foam. So this is a tensile test on an Auxetic polyurethane foam. Auxetic means that these structures are like deployable structures. So if you pull in one direction, instead of having a contraction in the transverse direction, as you have with conventional material, leading to a positive Poisson's ratio, when you pull, you have an extension in the transverse direction, leading to a negative Poisson's ratio. So we wanted to investigate how these auxetic effects developed in the material. So this is a very sort of cheap and cheerful in situ test rig, because these things are very soft, and so they're very easy to stretch by just manual pulling. The data was collected in Manchester. So this is just a view of going through the Z slices along the axis of the cylindrical foam sample. The bright bit is the cell wall, and the dark bit is just air in the pores. DVC was performed by Dave Hollis from La Vision, and after a noise study, we converged into a 48 cube voxel subset. And so from that, what you could get is the strain distribution, the transverse strain distribution. So that's the horizontal one when you pull vertically, as a function of the slice number. So this video here shows different slices as we're moving down the specimen axis. And what we can see here is that the strain values are dominantly positive, so in the sort of red and orange, with some pockets of blue. Just to make sure that this wasn't noise, this is a noise study based on rigid body motion, so we're well above the noise threshold. So what we're capturing here is some sort of signal, and showing that you seem to have a locally, at least, auxetic behavior. And then you can calculate Poisson's ratio globally on each slice, and reconstruct an evolution of Poisson's ratio along the specimen length. And that's interesting, but the big question with that is it's still pretty much qualitative. We were not able really to put any error bars on this. And we don't really know what this macroscopic Poisson's ratio really means when you have such a heterogeneous microstructure. And I'm going to comment on this later. A second example was trying to study bone implantation using an analog. So an experiment where we had an aluminum pusher pushing into a cylindrical specimen of saw bone which had a hole drilled in it with a slightly lower diameter than the pusher, trying to simulate bone implantation for hip implants. That was the PhD of Alex Martyr at the University of Southampton. So here are different slices of images of this implantation. So you see the pusher at different level inside the saw bone cylinder. And then we can get from DVC strains, which show, for instance, here between step 1 and 2, you can see a local compression here in the radial direction, coming from the fact that the pusher is pushing against the saw bone at the interface. And as you push this pusher further down, then you see that its hotspot is gradually moving downwards. So here, this is incremental correlation. So we take image n-1 and correlate it with image n. And this is interesting. I mean, qualitatively, we could compare it with a finite element model, which is what Alex Martyr did, using bone as a homogeneous equivalent material with a modulus and a Poisson's ratio. And basically, we captured a trend, right? There was a local compression, but the problem is, what do we do with this? Does that validate the model? The values are different. They capture the features, but again, no error bars, no idea what the bias is. And so it's only really qualitative. We also did another study to obtain material properties on trabecular bone, on real bone this time. So that was the PhD of Faye Gillard was early days, 2014, and I was involved in the later stage of her PhD. So she was working on porcine trabecular bone. This is a view of slices at different levels of compression. There was 6 or 7 load steps. And again, DVC was performed based on the subset that is schematically represented here, so containing a few trabeculae. And from that, you can get the deformation map. What was interesting first is to notice that the strain state was not uniform at all. So in this compression test, we were expecting that in a horizontal plane, the compression strain would be uniform, but it wasn't. And that's because it's very difficult to have a compression test without any kind of platen rotation or artifacts of contact between the bone and the platen. And this leads to some slight rotations, which certainly early in the test has a tendency to generate non-uniform deformations. And then we try to identify Poisson's ratio from this with 2 approaches. The uniform stress approach, considering that the stress is uniform, and that unfortunately has a bias, which was confirmed by simulation. So you see a sort of erratic evolution of Poisson's ratio. While by using a different technique, an inverse technique called the virtual fields method, we were able to see the gradual reduction of Poisson's ratio as the compression progresses, which is typical of cellular materials. So this is all very nice, but really, in a way, it's still quite quantitative. We're not quite sure exactly what we're measuring. And I'm going to explain why by moving into these DVC curves. So before and after deformation, this is what the material will look like. So this is taken from Alex Marter's PhD. You can see the implant, the aluminium here, and the trabecular bone phantom here, bone, which is a foam. And this is, you see, you haven't got contact here with the pusher and the material. And then after contact is made by pushing the pusher in, you could see that you have a local deformation of the foam close to the pusher. If you zoom in, this is what reality looks like before deformation, and this is what it looks like after deformation. And you can clearly see that this part here of the cell wall of that pore has been flattened, but the rest hasn't actually deformed very much. So very localized deformation at the level of the microstructure. Now, when you're doing digital volume correlation, you're using continuous shape functions, meaning that what you're trying to match is something that will look like this, a continuous deformation of that cell, and you're trying to match that with that, which cannot be described by a continuous function at that scale. As a consequence, you're going to have a mismatch, which will lead to a displacement bias. This also explains why you generally have rather poor correlation coefficients when you're doing digital volume correlation, because you're trying to match something that is discrete onto something that's continuous. The problem is also about the scale interactions. So not only are you going to have a bias, but also there's a more fundamental question about what does it actually mean. So this is a map, a strain map I got from that paper by Gianluca Tozzi and co-workers. It was done using DVC. You can see at the back the structure of the trabecular bone and overlaid on it a strain map. If you zoom in, you can see here strain contours, and clearly you see the underlying mesostructure. Now, strain, as it's defined here, is a continuum model. And you're trying to apply that to something that's fundamentally discrete here. And the question is, what do these strains mean? What kind of average do they represent? And we don't really have the answer to that question. In mechanical testing, the thing we like to have is scale separation. So we'd like to either resolve the heterogeneities, like in that case, or we would like to be sufficiently far away from the heterogeneities so we can treat this as a continuum equivalent. This is where we sit with that test, and that's the wrong scale. Unfortunately, while in DIC you can tune your pattern to scale, for instance, if you're interested in the strain, the grains of a metal at the scale of, say, 10 micron, then you can use gold nanoparticles under an SEM and image at very high magnification the strains directly in a grain. On the other hand, if you're not bothered about this and you want to look at a big structure, then you can apply a big pattern and completely ignore the microstructure and the mesostructure. And so in both cases, you completely separated your scales. Unfortunately, in DVC, you cannot do this. So you are caught up by what I call the DVC curse, because it forces you to work at the wrong scale. And that's because you're using the macrostructure as the pattern, as opposed as dissociating the structure with the pattern. It is possible to seed foam, for instance, with or polymers with gold particles, or try to have an artificial patterning in 3D, but it's quite complex and generally doesn't work. Certainly on trabecular bone, you wouldn't be able to do that. And so you have to live with the DVC curse. Now, how do you go around that curse? How do you exercise it? Well, there are two main ways of doing this, according to me. Either you keep the DVC concept, but you adapt the shape functions. So this is an example here. It's a crack propagation in a so-called Penrose structure. So this is a 2D structure realized by laser cutting, and it has this fancy structure to try to optimize crack energy absorption. So this is work done by colleagues at the École Centrale de Nantes in France, who were kind enough to share the data with me. This is a video of what happens. So you see, in terms of scales, it looks pretty much like what I've described with the DVC data that I showed before. It's the same kind of scale. It's the wrong scale, but this is the scale of interest. And so how do they tackle that problem? Well, they first make this shape by laser cutting from the CAD model, and then they paint it. So they have an experimental artifact, and you can see the little dots here corresponding to the speckle pattern. And then they create a synthetic image based on the CAD model. They then have to register the magnification and the location. So they do this by a global correlation problem over the whole field of view, just as an initial starting point. Then they perform an FE-based DIC just to capture the load, the global deformation patterns. And once they have that, they use that data to initiate a more local matching based on beams this time. So what they're using is they're using beam kinematics as shape function in the DIC problem. And that's quite interesting because you can then, let me just play that video. You can again define shape functions as beam shape functions. So each node here has two displacements in plane and a rotation. They used here cubic shape function for the shape of the beam. This little video shows the initial node position. So you see here this sort of star shape that was before deformation. The image at the back is the image after the test, and you can see how the nodes gradually follow the deformation through the test to match the deformed position. Now, once you have that, you can then calculate the energy in the beam, and they were able to calculate the strain energy absorbed during the test. And they did different observation on different type of geometry here, but I don't really have much time to elaborate, so I'll just keep it. So you can adapt your shape function. Or the other thing you can do is to go through a finite element simulation to perform what I call finite element assisted DVC. So in that case, I got data from that paper. So it was data on polyurethane foam. You could see a scan here of the foam, very similar to what I showed before. What they did is they extracted that to a CAD model, which is fairly standard these days, and then to an FE model. So this is all off-the-shelf technology. Now they did a finite element model. They tested the material properties on a separate material test, and I think that's the weakness of that approach here. Produced finite element data, and then they're trying to map the load, the global load versus displacement, which is a very integrative measure, but in any case, that was the point. But you could do something differently here. What you could do is use the same approach, scan CAD model, FE model first. Then with initial material parameters, you can perform a finite element model. From that, you get a displacement map. And from your initial image now, what you can do is numerically deform this image map, this initial image, to produce a synthetic deformed image. You can then compare that to your initial experimental image, build up a cost function which is the difference of the two, update your material parameters until these two match. And that will provide you with the material parameters of the constituent of your material. You can acquire images either in the volume if you have time. You can do projections if you want to go fast, because you don't need to resolve the full field now. You just need projections, or even 2D image might be enough if you want to go really, really, really fast. Advantages of drawbacks of the two approaches. I think the main advantage of the kinematic one here is that you don't need material properties. The big disadvantage of the other one is that you need material properties. On the other hand, if that's what you're after, then this is the right method, because I think the higher order shape function would not really be able to give you that. A brief conclusion on what I've described here today. So the message is that DVC as it stands, sort of the share of digital volume correlation, gives you some sort of blurred information. And it's quite qualitative. It gives you some sort of strain average that is not well defined mechanically. I think it is a bit of a problem. Alternative approaches are still to be developed. There's no commercial tool, but I think it's something that is very interesting to develop in the near future. And a killer application would typically be high strain rate properties of trabecular bone or cranial bone, where the structure, I mean, the material in itself doesn't exist. It comes with the structure. Right, thank you for your attention. I will stop here.